Hello, everyone. I'm Nalan Özkurt. Today, we are making a graduate school seminar by Musa Tanimu. He will present us deep learning for EEG-based biometric systems, and he will talk about the basic biometric systems and his PhD thesis. Musa, you can start whenever you are ready.
Okay, Hujam, thank you very much for the introduction. My name is Musa Bindawa Tanimu. I'm a PhD student at the Department of Electrical and Electronics Engineering at Cheshire University. I'm at the same time working on the TÜBİTAK 2244 Industrial PhD program with EDS Company and Nalan Hoca, Mustafa Seçmen Hoca, Junaid Hoca, and two of my colleagues. Today, I'm going to talk about deep learning for EEG-based biometric systems. The outline of my presentation is going to be, I'm going to give a brief introduction about EEG biometrics and EEG-based biometrics. Then I will talk about some technical backgrounds of the deep learning, general, and the EEG data collection. Then I will show you our experiments that we've done so far and the results, and I'll briefly talk about our plans for our future work. So I'm going to start with biometrics. Biometrics are measurements based on the unique physical, physiological, and behavioral characteristics of a person. The types of biometrics are like we have iris, face ID, we have fingerprints and brainwave biometrics, etc. Biometrics is a very old field because the first known fingerprint was recorded by farmers. It dates back to the 14th century. So biometrics has been in use for quite a long time. Any characteristic that is unique, universal, stable, and collectible from an individual can be considered a biometric system. By unique, it means that a biometric from an individual is different from the biometric from every other individual. By universal, we mean that the biometric is available, generally it is available in every person all around the world. By stable, we mean that if we collect the data, the biometric data over a long period of time, does not change in a way that is noticeable. And by collectible, we mean that this biometric is easy to acquire and store. Biometrics are commonly used for military and security operations. We use biometrics for person identification and person authentication. By person identification, we mean that we can use biometrics to identify one group of person from a group of one person from a group of person that we already have their data in a database, while from authentication, we mean that we are able to verify whether a person is who he claims to be or not. Research shows that EEG signals carry genetically specific information unique to individuals. As I mentioned earlier, the uniqueness of biometrics is important. So this EEG signals having this specialty is important in the field of biometrics. So now I'm going to talk a little bit about EEG. The processing of all the information carried and sent throughout the human nervous system is done in the brain. Our brain consists of 86 billion neurons that are activated with energy whenever we think or move a muscle. EEG is the most commonly used data collection method for brain energy signals. EEG research commonly focuses on medical applications generally, but there is growing interest in the field of security. This is where EEG-based biometrics comes in place. EEG is a good method for biometrics because it is non-invasive, which means like we don't have to cause any damage to the person's head or brain before we can collect the signal. Therefore, the signal is collected and stored easily. EEG is not easy to forge, and it is almost impossible to collect without the person's knowledge. EEG has a clear advantage over other methods because it is universal, and because of its universality and its uniqueness. It can easily be collected from persons with disabilities. Unlike other biometric systems, for example, you cannot collect like the iris data of a blind person, and someone with like hand disability, you cannot collect their fingerprints easily. However, EEG, with the EEG devices, you can collect it from other people. So this is a very good advantage of EEG. However, like EEG has time variability and permanence problems. So this is a disadvantage of EEG, which can be studied and improved on like based on research. This is one of the things we plan on doing in our research. So the collection of EEG signals, EEG signal is measured from the current that flows during the transfer of information on the nerves that are activated in brain cells. The current generates an electric field over the scalp, which is measured by the EEG. It also generates a magnetic field, which is recorded by the EMG, but that is not very important for us. For us, it can be considered as noise. Different parts of the brain have different roles, hence they generate different signals depending on the brain activities. According to this, the electrodes from EEG devices are placed on different parts of the brain in order to collect these signals. The electrode scalp placement is very important in getting high quality EEG signals. The type of activity that is critical for EEG biometric systems depends on the activity that is being done by the person which the EEG is recorded on, and depending on where the electrode is placed. The general acceptable standard of the placement of electrodes is the 10-20 system. The 10-20 system is the system whereby 10 to 20% of the front to back or left to right part of the scalp, the electrodes are generated with that gap, the 10 to 20 gap accordingly. Data can be collected when an individual is resting or doing sensory tasks or cognitive tasks. The recordings can be taken in sessions separated by a chosen time for various reasons in controlled environments to limit external noise. The resting state is good for biometric identification, because when a person is resting, he's in a relaxed position, he's not doing any action, he or she has their eyes closed or open, but usually for biometric purposes, it is done with the eyes closed. However, like from literature, you can tell that cognitive stimuli, which is more repeatable actions, have better information about individuals. But the disadvantage of this cognitive stimuli is that it takes a lot of time. So for the purpose of our study, so far we've been working with the resting state data. Now I want to give a little information about deep learning. Artificial intelligence is a branch of computer science that creates systems which mimic the human brain. Machine learning is a branch of artificial intelligence where computers learn and improve from given data and previous observations to solve problems using algorithms without the need to be told what to do by human intelligence. Most common applications are classification and regression problems. Artificial neural networks are classification tools used for solving problems. ANNs consist of multiple fully connected layers of neurons. They are good for nonlinear problems. As you can see on the image on the right, these neurons are interconnected from the input to the hidden to the output layer. Deep learning is a branch of machine learning based on deep artificial neural networks. By deep, we mean they have more layers than regular artificial neural networks. Deep learning is focused on representations of learning data. Deep learning is good for image and speech recognition system and also natural language processing. Convolutional neural networks are a type of artificial neural network designed to break down inputs into smaller and more manageable parts called features. The features are fed into a series of layers where complex calculations to recognize patterns are formed. One of the most important components of the CNN, which it derives its name from, is the convolutional layer, where filters are applied to the input to extract specific features. Another important component is the pooling layer, which downsamples the features to reduce the network's computational complexity. CNNs are widely used for image and visual data applications, but in our case, we use it for signals. So now I want to talk about some of the experiments we've done and also discuss the results. Of the first experiment we did was using the Colorado State data set, which was taken from 4 subjects. They were given 5 tasks to use. These tasks are the baseline task, which in our case is the resting state, multiplication, letter task, rotation task, and the counting task. But for our own case, since we are going to use it for biometric purposes, we chose to use the baseline task only, which is recorded at the resting state. The data is a 10 seconds data of 10 trials each at the 250 Hz sampling rate. The data was collected from 6 channels, the C3, C4, P3, P4, O1, and O2 channels. We decided to do some windowing to expand our data. We chose the 1 second, which is 250 Hz data points for our windowing size, and we slid it all over the signals at 25 data points until the end. In the end, we got a 6 by 250 by 90 matrices from each trial for each subject. That is for 4 of the subjects, 10 trials each. We have 6 by 250 by 90 matrices. And this is just an example of the signal plotted. So we fed it into a CNN with this structure. At the input layer, our input size is 6 by 50. At the convolutional layer, we had 10 1 by 50 filters. At the max pooling layer, this is the size of the pooling filter. Then comes a second convolutional layer of 1 by 50 size, but this time we had 20 filters, followed by a max pooling layer, then another convolutional layer, then a fully connected layer. And after every convolutional layer, we had a batch normalization and ReLU layer. And at the end of the fully connected layer, we had a softmax layer. And we trained our network with the stochastic gradient descent with momentum with the initial learning rate of 0.1. And all these parameters, we got them by trial and error. And at the end of our experiment, at first we tried to feed as inputs channel by channel. Since we had 6 channels, we fed the data of each of the 4 subjects. The first channel, we got our accuracy was 86.5%, and the required time was 18 seconds, etc. Until the 6th channel, we did it accordingly. Then we decided to feed all the channels together, and we got higher accuracy, but it took a little bit longer computational time. So looking at this, we can tell that some channels give higher accuracy, as we discussed earlier from the literature, like some, depending on the activity, like some specific parts of the brain work better for some activities. Like we get better, higher quality data from some electrodes, depending on the activity doing, and we can see from the accuracy gotten here. Then we decided to try something else. Because this one, we fed the raw data into our network. So we decided to try maybe applying some pre-processing. We decided to pre-process our signal by doing some wavelet transforms, then feeding it into our system. You can see the results when we feed the raw data. We have the train and test accuracy here and the required time of 16 seconds. When we use the mollets, accuracy drops and the required time increases. When we use the complex mollets, like our accuracy is still lower than when we feed the raw signal, but the required time is higher. But when we use the discrete wavelet, we have a much better accuracy. However, we can see that the required time increases. Then we decided to, from the information gotten from this previous experiment, the problem with the previous experiment is that our data set is not very big, like it is a small data set. So we decided to look for a better data set to test our network and see the results. So we found the BED data set, which is the biometric EEG data set. It is a new data set specially designed for EEG-based biometric work. This data set is collected from 21 healthy subjects, and they were subjected to 4 different stimuli. And the EEG signals were collected in 3 different sessions, separated by 1 week. The data was collected via 14 channels, but for the purpose of our experiment, we used both the 14 channels, and then we decided to drop the number of channels to 4. Like, we decided to drop the number of channels to 4. We chose these 4 channels based on the channels with the higher accuracy from our previous experiments, and see whether our accuracy will improve or not. Because one of the aims of our study is to reach an optimal number of channels for EEG devices in the future. So this is our network structure, which is a CNN network structure. It has the convolutional layer of filter size is 2 by 50 of 10 filters, followed by batch normalization and ReLU layers with the max pooling of size 1 by 10 of 2 stride. Then the next layer is shown like on the graph, followed by the next layer by the next layer. And we also train it with stochastic gradient descent with momentum with initial learning rate of 0.01. The metrics we used are correct classification rates and F1 score. And this is how each of them were calculated. And this table shows the results of our experiments with the 14 channel input. We used the K-fold cross-validation for training our network. We used 5 folds, and we used 80% of the data for training and 20% for testing. And our correct classification ratio, when we fed the 14 channels, was 72%, and the time elapsed was 626 seconds. And when we decided to drop our number of channels, we chose the channels with the highest accuracy from our previous experiments, and we decided to use it as input. Our accuracy increases to 83.51%, and the time elapsed is reduced by almost half, which shows that reducing the number of channels might work to our advantage. And this is also a comparison of our work with existing works in literature. And it is fair to say that most of this, from this literature, most of the work, we use the same data set, we use the BED data set. However, some of them used the resting state like us, while others used other tasks. And you can see that our study below has significantly high correct classification ratio in comparison with some of these ratios. And we fed our system with like the raw EEG signal. Meanwhile, most of the other work, they did some pre-processing and fed the features from this pre-processing into their own chosen networks. And for the future, we're planning on using recordings of optimal channels for biometric purposes, and maybe using other tasks like the cognitive task, because like I mentioned earlier in the earlier slides, it is more consistent because you're asking a user to do some task repeatedly, so it is easier to get the same or similar EEGs, so it can be used better for identifying individual. However, it takes longer and the computational complexity is much. And we can try different deep learning architectures to make best use of the time variability of these signals. And one of those architectures we're planning to use is the RTPNN, the Recurrent Trend Predictive Neural Network, which is good for time variant signals. This is the end of my presentation. Any questions?
Thank you, Musa, for this presentation. And we are also like to thank all the attendees for their attendance and their questions. Thank you all. See you in other graduate school seminars.
Thank
Thank you,
all.
Musa. It
Thank
was
you.
a nice seminar.
Thank you, Musa. Thank you very much.
Thank you.
Thank you, Musa.
Thank you for listening. Thank you for the questions.